Well, welcome everybody. It's a great pleasure to have you all here at Gladstone today. I think most of you know who I am, but my name is Deepak Srivastava, and this is the first President Council Symposium that I'm leading as the new president of Gladstone. I'm delighted to see so many of you here, old friends and many new friends. Before we start, I want to acknowledge the great privilege that I have to lead this organization, which is really one of the gems of the world, and in particular to recognize Bob Maley, who's our founder of Gladstone and our first president, and Sandy Williams, who's served as the president for the last 8 years, and has left this organization in really great shape and in a position to really launch to the next level in trying to solve some of mankind's worst diseases. And I also want to acknowledge Bill Price, who's one of our 3 Gladstone trustees, who's with us here also today and has been a great supporter. So, I want to start off by talking about why we're all here. And by we, I mean, why are all the employees at Gladstone here? Why are you all here, which are our friends and our supporters? And I think it's really quite simple. We're all here because even though we're now sit here in 2018, so many human diseases still have no solutions. They're intractable diseases, and you all have your loved ones and friends who I'm sure are suffering from the same. As I'm standing here right now, my wife is in the hospital at UCSF with a neurodegenerative condition that has no cure. My father's in the hospital in Texas with heart failure, which also has no cure, and that's just not okay. And so it's not okay for us to be here and not be driving as hard and as fast as we can because there's urgency to this. It matters to people. And so that's why we're all here. I'm sure that's why you all are taking time out of your day to be here and learn, and many of you have supported us quite generously, and that's what's really allowing us to do the things that we're doing. And so as I started as president in January, we had an opportunity to step back and evaluate where we are. And the bad news is that so many human diseases are still intractable. The good news is that we are sitting in an age where we finally have both the knowledge base upon which to understand human disease at a level we haven't before. We have access to human cells like we've never had before to figure out why people get disease. And we have the ability now to alter our genomes. So we can read the code, the genetic code. We can interpret the genetic code. And what you'll hear today in the program is a bit about how we're using artificial intelligence to interpret the code. And finally, we can write the code. Some of you are familiar with the term CRISPR. It's a new gene editing approach pioneered by Jennifer Doudna across the Bay that allows us now to write the code. And so that's the good news. And how do we get there? And so that's what I want to spend a few minutes because this is the first symposium where, since we've had this transition, I thought I'd spend a few minutes sharing with you our vision going forward before we launch into our program today. So we have great momentum in the areas of cardiovascular disease and neurodegenerative diseases. Gladstone has made major investments over the last 2-3 decades or longer in those areas, and we're finally at the point where we understand more deeply those diseases. We're beginning to see therapeutics launch into companies that are taking those into clinical trials. And so we're going to keep pushing hard on those and build on our momentum. In the area of viruses and immunology, where we've also made a big investment, particularly around AIDS, where we've been at the epicenter of one of medicine's greatest success stories of taking AIDS from a universally deadly disease 25 years ago to a chronic disease. We have reason to be really satisfied with that huge transition. But we're at a position now where we can turn our attention to the next big challenge. And we intend to build on the paradigm that has been established for immuno-oncology that many of you also must be familiar with, where we've been able to harness our own immune system in ways we never could before and engineer it so that it can kill cancer cells throughout your body. It is a huge revolution going on in oncology and cancer right now. That same paradigm of engineering and harnessing your own immune system could be leveraged for infectious diseases, for autoimmune diseases, and for inflammation all throughout our body. So we're intending to make investments in that area, which I think can affect so many human diseases by fundamentally understanding the human immune system in ways we never have before. Now, what is driving all of this and the pace, the accelerated pace of discovery in each of these areas? It's pretty simple. It's technology. There have been huge advances in our ability to develop and use technologies to interrogate human cells in ways we never could before. So what's the net result of all that technology? You get a lot of data. When you get a lot of data, there are two things that could happen. You could get awash in it and drown in it, or you could make sense of it. And what we've decided to do here, and we had been over the last several years making investments in both developing new technologies at the real frontiers of those technologies, and making investments to be able to analyze those data, write new code in creative ways to make sense of it. And so recognizing that this is going to be a major driver of biology going forward, we recently launched, in January after I started, a new effort at Gladstone that we've called the Institute for Data Sciences and Biotechnology. And Katherine Pollard, a computational biologist who's been here for the last 10 years, has emerged as one of the international leaders in this field, is the new director of that. And you're going to hear more from Katie in a moment around this program. So that's how we're going about tackling human diseases, leveraging the most modern approaches and technologies and data analysis to tackle those diseases. So what I'd say is that we have these challenges where we can't address human disease, but we have the greatest opportunity that we've ever had to actually make a difference. And the model that we've developed at Gladstone to build focused teams around a few major problems, we feel that we can really tackle these and make a huge difference in human suffering. So with that, I wanted to share that with you because many of you have spent your time and monies supporting us for many years, and I wanted you to be clear on where we're headed as an institution going forward. So now to our program today, which is a really exciting one that's very current. You guys hear about artificial intelligence all the time. I'm going to invite Katherine Pollard to the stage now, who's, as I mentioned, is directing this new effort. Katie is one of the world's leaders in computational biology in many different areas, including the microbiome. And she's a wonderful teacher as well, as you'll see. And so, Katie, if you would, please come up and frame our program for the audience.
I'm so excited to be directing this new effort at Gladstone and to be here with you guys tonight. I am going to do a little teaching here at the beginning and start by asking, what is artificial intelligence, or AI? What are we actually talking about here tonight? This term probably conjures up ideas and images of computers beating people at chess, for example, or the image recognition software on your phone that can tell whether the snapshot you just took is your sister or your son. And so we all experience and hear about artificial intelligence, and what we're going to do tonight is talk about exactly what it is, break it down a little bit, and then discuss how through some great examples, how we're using it in biomedical research. So that's our agenda. So what is AI, or artificial intelligence? Fundamentally, it's computer code, and it's code with a purpose. Two things. One, finding patterns in data, and optionally, number two, using those patterns to make smart decisions. So what do I mean exactly by that? So let's start with learning patterns. and let's talk about what the data is. We're going to have, just to start with, really simply, 2 objects. And for each of those objects, we're going to measure a bunch of things about them. So if those are 2 faces that you've just snapped pictures of, the objects are the faces, the pictures with the faces, and these features or characteristics that we're measuring on them could be the intensity of each pixel in that image. Now, in biomedical research, maybe the 2 objects are 2 cells. And what we might measure is the level of, say, proteins in those cells, either with a genomics experiment or with imaging. And so then we have lots of proteins, thousands of proteins, and for each cell I have levels for those proteins. It's not so different than those pixels in the 2 pictures, right? Conceptually, I've got objects, and for each object I've measured a lot of things. And what the first task is, the pattern recognition task, is asking whether these 2 things are the same or similar to each other or not based on all this data I have on them. So are these cells the same kind of cell? And by that I mean, are the levels of their proteins similar enough that I would say that they are close to each other, or they're not so close to each other? So that's pattern recognition. And then the second task of using those patterns to make smart decisions is a prediction problem. So the first part, pattern recognition, we also call that unsupervised learning or unsupervised machine learning. Trained as a statistician, I call it clustering. Those words all mean the same thing. It's finding patterns without even knowing what we're looking for. So the second thing that you might do is to take patterns and put labels on them, like this picture is my sister, this picture is my son. And then when you take a new picture, the task could be to try to guess or label that as your sister or your son and put it into the right folder on your phone. Or with cells, we might say, well, this is a dying cell and this is a healthy cell. Or this is a newborn cell and this is an old cell. Or this is a cell from one kind of tissue or another kind of tissue. And so we can learn those patterns, and that's important in biology right now because we don't even really know in this high-dimensional data what the types even are. So that's the pattern recognition part. The prediction part would be when I see a new one and I don't know what it is, can I predict that it's old or young or dying or not, just from the data on it, and some examples of the kind of thing I'm looking for. So that's called supervised learning. Supervised because you've given it some examples of the outcome or the prediction that you want to make. And the same input data then can lead to predictions on examples where you don't know that outcome or that label. So just to review, there's 2 tasks in AI. One is just learning patterns, unsupervised, describing data, what kind of patterns are there, what kind of things are similar to each other or not. And then the second kind of task, supervised learning or prediction, is using those patterns and a few examples where you have labels of some outcome or type and trying to make a prediction. So what's new then? These are old concepts. I learned these ideas more than 15 years ago in graduate school. These are just the models that we've always fit in statistics. So why is there all this hype? What's going on with AI right now? So I would say there's at least 3 things that have happened recently that are exciting and that make this exciting for biomedical research specifically. One is that the data sets are just much bigger. Number 2, there's some new algorithms that are really interesting and effective. And number 3, which I think is the most important for us here at Gladstone, is there are new and emerging methods for not only doing a good job at these tasks, but also understanding why we do a good job at that task. And that's actually fundamentally what we care about here in basic science. It's not so much like labeling cells as we see them go through a sequencer or through a microscope. It's knowing what makes a cell different from another kind of cell. What about it means that it's dying or about to die, as you'll hear about tonight. So let me just give a quick example from my own research to demonstrate those 3 important things about why AI now. Why big data? So in my own lab, we're working on a problem, a huge problem in human genetics, which is that most of the disease associations, the mutations that we know are related to diseases, are not in genes. Most of them, especially for the complex diseases like neurodegenerative disease and cardiovascular disease that we study here at Gladstone, most of those mutations are not in a gene. And if you look in the human genome, most of the genome is not genes. And when you see one of those mutations, there's a lot of genes nearby that could be the one that's affected. But we don't know which one it is because we don't understand how mutations cause genes or pathways in cells to go awry. We just don't have that knowledge yet. So the way we're using AI is to get lots of data about cells and about these positions in the genome, everything that we know about that position in the genome, and then we're trying to not just learn those patterns, but perform the second task of prediction and say, can I map that mutation onto a gene or a pathway or a process that's going to actually teach me something about the disease? So big data is important there because we need a lot of examples to be able to do that accurately. And there's no one annotation or piece of information about this mutation that will really predict. Each one is very bad at predicting, but if we measure a lot of things about that mutation, about that part of the genome, and about all the genes, if we measure enough things, we can accurately make those mappings. And it's only possible with the big data because you need complex relationships. And to learn those complex relationships, it's not like yes or no, it's this or that, but if not that, then maybe this. But if not that, then this other thing. And that whole pattern would say, oh, okay, yeah, it goes over to this gene over there. And so those kind of complicated, not just yes, no, things need a complicated algorithm. And there's some new algorithms or new versions of old algorithms that are very popular right now and very good at learning those complicated relationships that aren't just like this happened and then that happened. And then most importantly for me, what I wasn't so excited that we could necessarily map these mutations to genes. That was very cool. But we asked which of the many thousands of pieces of data that we fed the computer are helping us do that. And then what that showed us was that there were things that the computer had learned about biology that we didn't know. So the computer, in showing us how it worked and how it solved this problem, taught us something. So in this example, we learned some proteins that when they bind nearby a mutation really indicate which gene it's going on to regulate or which process is involved in the disease. And those patterns are not one protein. They're actually complexes of proteins. And so going back to the image recognition problem, the analogy is that these new AI algorithms, they don't just tell you like one pixel in that image is going to say that this is your sister and not your son. There's no single pixel that can differentiate those two people from each other. But there are features, combinations of pixels, that encode something like the shape of an eyebrow. And that does differentiate your sister from your son. And so we're trying to find the biological equivalent of these eyebrows, basically. And they're these combinations of very complex combinations. And when we find an eyebrow, the equivalent of an eyebrow, we learn about how proteins work together and how they cause disease. And so this last feature, this ability to use the algorithms to actually learn about mechanism, is why I'm most excited about AI and how we can use it in biomedicine. So tonight, we're going to hear a couple of other great stories about that paradigm from two Jeremys. First, you'll hear from Jeremy Linsley, who's in the Finkbeiner lab here at Gladstone. And second, from my colleague at the Chan Zuckerberg Initiative, Jeremy Freeman. And then I'm going to get up at the end after you hear a bunch of good news, and I'm going to tell you some bad news about why these approaches are not perfect and where the pain points are and what we're working on in the future. So let me welcome the first of the Jeremys to the stage.
Hi. Get this adjusted. Hi, so I'm Jeremy Linsley. I'm a postdoc in Steve Finkbeiner's lab at the Gladstone Institutes. Today I'm going to talk to you about how we use a microscope that's controlled by a robot and combine that with artificial intelligence to understand the mechanisms and develop treatments for neurodegenerative diseases. So first I want to level with you. I'm actually trained as a biologist. I study cells and neurons and model organisms under a microscope. a couple years ago, artificial intelligence was just a fancy new sounding technology that I thought was more science fiction than real science. But as I've had a chance to work with it in the lab and use it on my research, I'm totally hooked. It's changing how we do experiments in the lab. It's changing how we interpret our results. It's changing how we think of neuroscience. So I hope right now I can tell you about how this is transforming how we do research in the lab. So in the Finkbeiner lab, we focus on neurodegenerative disease research. Between Alzheimer's disease, dementia, Parkinson's disease, Huntington's disease, and ALS, or Lou Gehrig's disease, they combine to make the #5 killer of people in the United States. But what's worse is actually, as probably most people in the room have had experiences with people either affected or suffering from these diseases, we know that the emotional and social toll of these diseases is really bad. These are really devastating diseases. But what's really scary about these diseases is that currently there is no disease-modifying therapy for any of these major neurodegenerative diseases. But that's not for lack of trying. There's major research initiatives across the planet looking at the mechanisms and trying to develop treatments for these diseases. There's great organizations that fundraise money for funding this research and for raising awareness of these diseases. And there's even pharmaceutical companies have invested heavily in pushing drugs through clinical trials. But at this point, none of these drugs have succeeded, and we still don't have any cures for any of these diseases. So why is that? So I want to start by telling you a few reasons why neurodegenerative diseases are especially hard to study. So for one thing, most neurodegenerative diseases are late onset. So you can really live late into adulthood before showing any symptoms of these diseases. To add to that, many people show onset of diseases at different times. We don't understand why some people are getting it earlier than others, for the most part. And then on top of that, clinicians report that many people who start to show when they start to show neurodegenerative disease symptoms, it's well beyond, the disease has progressed well throughout the brain, making it even harder to treat. So we're really working with a moving target here. To add to that, some neurons are affected while others are not. On top of all of this, many of the neurodegenerative diseases are characterized by neuronal death within the brain. And once we lose neurons within our brain, they don't regenerate. So we also have the problem where we need to treat these diseases before we get massive cell death within the brain. Add to that, the brain is extremely complex and completely inaccessible. What we really want to be able to do is cut open a person's brain and dig through all the neurons and non-neuronal cells in the brain and find the ones that are unhealthy and find the ones that are healthy and treat just the ones that are unhealthy. As you can guess, as you can imagine, that's really impractical. But even if we were to be able to go in and pick out the specific neurons that we want to treat, at this point we can't even tell which ones are unhealthy and which ones are not. So in the Finkbeiner lab, we work to study these unhealthy neurons from these major neurodegenerative diseases in our lab. And we do this by using stem cell technology, which was developed in part here at Gladstone Institutes, that allows us to take neurons, grow neurons from patients with neurodegenerative disease in the lab, and allow us to watch those neurons in the lab. And then to add to that, we use a technology that the Finkbeiner lab invented we call robotic microscopy. And this allows us to rapidly and automatically image these neurons with neurodegenerative diseases over long periods of time so that we can really watch the progression of the disease in the lab and then potentially develop treatments for these diseases. This is where artificial intelligence makes a huge difference. So first of all, our robotic microscope generates millions and millions and millions of images. And these are actually hard to analyze. So what we've actually done in the past is we've hired undergraduate interns to sit in front of a computer and manually annotate a neuron over long periods of time. And so this is not only, it's cost prohibitive. It costs about $1,000 for a college intern to do a single experiment. And it's also time consuming. It takes them sometimes about a month. And then there's another problem, and you're going to have to take my word for it here. College interns are not very good at this. But to be fair, even trained experts, it's really due to our human eye. Even trained experts are really only to pick the time point at which a neuron dies in our experiments with about 85% accuracy. So I developed a genetically encoded biosensor which can identify whether a neuron is alive or dead with 100% accuracy. But then we took data using that biosensor and trained a neural network to use artificial intelligence to pick in all of our samples whether a neuron is alive or dead. That algorithm is able to identify whether a neuron is alive or dead with over 95% accuracy now, and it likes to wake up before noon. So being able to precisely define when a neuron dies is really important for us. As I explained, we really want that critical time period when a neuron is sick but not dead. So using other algorithms we developed, we can now predict whether a neuron in our time lapse will die within the next 24 hours with up to 85% accuracy. So we think that this is going to really help us hone in on that specific time when we really want to be looking at these sick neurons and developing treatments. And then when we put our treatments on these neurons, we can tell whether we've affected the fate of that neuron. But I think the most exciting part of using artificial intelligence in our lab has actually been that we find that the computer is able to see more in our images than the human eye. So our microscopy images are actually very rich with information, but the problem is that it's really hard to extract that information and understand what we're looking at. So normally, conventionally, in microscopy, we understand our images by staining them and dyeing them and using antibodies to label specific parts of those images that we want to be looking at. The problem with this approach is that it constrains our information to a specific type of information. So for instance, if we're trying to look at the nuclei, we can stain our cells for the nuclei, but then we're only seeing nuclei in those images. Using multiple types of dyes and labels in combination is really problematic. Most of the time they cross-react, and we have only so many channels that we can look at once. Additionally, and more pertinent to neurodegenerative disease research, we found that when we put these labels on our cells, it actually makes them unhealthy. So it's hard to tell what's due to the neurodegenerative disease and what's due to the labels. So in an ongoing collaboration with Google that was actually spearheaded by Mike Frumkin, who's here in the audience somewhere, the ThinkBinder Lab just published a paper with Google showing that we can train artificial intelligence using conventional labeling methods to identify within an unlabeled image what the structures are within that image. So on the left, you can see an image of a bright field image where you see kind of clumps, but it's really hard to know what you're looking at there. And on the right side here, we have an in silico labeled that same image is in silico labeled. And you can see all the red and the blue and the yellow in that image are identifying features of those cells that when we use conventional labeling, we find the same thing. So we think that this non-invasive approach will tell us more about our neurodegenerative diseases, as well as show when our therapies are working or not working. So there's actually a lot more that the ThinkBinder Lab is working on, and I'd love to talk to you about this more, but I'm short on time right now. I just want to leave you with one thing that's really exciting to me in artificial intelligence, and that we're currently trying to work on, and we're currently pushing forward. So I've told you about our robotic microscopes that can produce data automatically, and I've told you about our algorithms which can analyze our data automatically. Another thing we can do with this is have the computer actually run the experiment completely on its own. So we can have the computer either administer drugs to our cells or control the neuronal activity, and then analyze that image and what it's just done to the cells, and then learn from what it's just done and continue the experiment. And this closed loop approach of artificial intelligence is called reinforcement learning, and it's a really powerful way of figuring out patterns in data. And as you've probably heard reports of AI beating the grand chess masters and AlphaGo masters, and this is the type of approach that they're taking. So I'm really excited to start using this, and we're trying to adapt this to understanding neurodegenerative diseases and really dissecting the complex characteristics of these diseases so that we can develop cures for these diseases. So with that, I'm going to pass the mic to Jeremy Freeman.
All right. Thanks so much. I'm Jeremy number 2. Thanks, other Jeremy. So I'm Jeremy Freeman, and I'm here from the Chan Zuckerberg Initiative. And this is a relatively new organization. We're a really unique organization because we're sort of 2 organizations in one. So on the one hand, we're a philanthropic group, and we are really trying to support biology and the scientific community through grant making. But within the same organization, we also have a computational biology and software engineering team that is working on building tools, algorithms, computational methods for doing a lot of the things we're talking about here, understanding data, processing data, handling data, and sort of finding patterns using new methods from computation and machine learning. And to me, this is an incredibly exciting organization to be in because it's trying to do these 2 things together and do them in a way that's maximally synergistic, so we can do things like give grants to experimental scientists that are solving really interesting new problems and collecting really cool data sets like the ones you've been hearing about in cell biology or neurodegeneration or other areas. And then within the same organization, we do computational biology and software engineering to understand the challenges they're facing, collaborate with these groups, think about new machine learning methods, new approaches to analyzing their data, and then work collaboratively with them to both understand those problems and hopefully come up with solutions. This model, to me personally, is super exciting because before I was here, I was running a neuroscience research lab at Genelia Research Campus, and I sort of witnessed firsthand just the challenges that scientists, especially modern biologists, face with data analysis and computation. As just one example, a group I was collaborating with and worked with, this guy Nick Safroniev, who was in the lab of Karl Svoboda. Nick's an incredible scientist, and he built this truly fascinating instrument and experimental setup. So he was interested in watching patterns of brain activity, measuring patterns of brain activity in awake behaving mice. And he built this incredibly large microscope. It was like 5 times the size of Nick, 100 times the size of the mouse. And the microscope was both the microscope as well as something called a tactile virtual reality environment. So it turns out when you're trying to measure brain activity in a mouse, you want the mouse to be stationary because the microscope is stationary, so the mouse can't literally be running around. So Nick built this incredible setup where there's a mouse in this environment with walls, and the walls move around the mouse to simulate the experience of being inside of a maze. And then the microscope on top of the mouse's head is measuring the activity of tens of thousands of neurons while the mouse is running around. So this allowed Nick to make measurements that no one had ever made before at a scale that was truly remarkable. And Nick spent a lot of his time looking at these images and drawing circles around neurons, because every time you capture one of these movies, you now have 10,000 neurons. And kind of just like Jeremy said, there's the task, which is to identify the groups of pixels that correspond to the individual neurons. And kind of in a nutshell, all the time I spent at Genelia, I just wanted to know, how do we let Nick do more experiments and make more measurements and not spend his time drawing circles around neurons? And that's true for neuroscience and for this domain. It's true across a lot of biology. One of my other former neuroscientist colleagues used to say, 10 years ago, the lab spent 80% of its time doing experiments and 20% of its time doing data analysis. And nowadays, it's the other way around. And it's 80% of the time is analysis and computation. Now, for someone like me who likes computation, that's great, that's wonderful. I love doing that. But what I really want to do is enable scientists and do and automate as much of the computation and analysis as possible so that people like Nick can just do more experiments and make new discoveries. So that's a lot of what we're trying to think about at the Chan Zuckerberg Initiative. We work closely with labs on the ground to understand their computational problems and come up with solutions, and then make those solutions openly available to all researchers. So everything we do is either making data openly available or developing open source tools and technologies. The particular project I wanted to highlight today and just tell you a little bit about is a project that we're involved in supporting at Chan Zuckerberg called the Human Cell Atlas. So this is a really exciting started as a grassroots effort that now has about hundreds of labs involved. And this is an effort to map, systematically characterize all the cells in the human body. So your body has trillions of cells, and one of the things that's kind of crazy is that every cell in your body more or less has the same DNA, and yet all of your cells end up doing completely different things. cells in your brain are very different than cells in your skin, and they're very different than cells in your muscles. And that's because of how genes are expressed, and then ultimately the proteins that form. And you can measure that to some extent through methods like RNA sequencing, where you're characterizing patterns of gene expression at very high resolution and very high throughput. And the technology for this has done sort of amazingly been transformed and increased in scale and throughput over the last, even just last 5 to 10 years. And the fact that the cells in your body do different things is really important, because it's how our bodies do all the incredible things that we do. And it's also important for basic understanding of health and biology. It's also important for disease. You can think about the fact that the cells in my muscle, well, not me, I don't really work out that much, but for people that are really strong, the cells in your muscles, like your arm muscles, they regrow. So when you go to the gym, you work out, cells break, and then they get stronger. Again, for some of you. The cells in your heart don't do that. They don't regenerate that way. And maybe if we knew the difference between the two and why they were different, that would be helpful for understanding how to make a heart cell a little more like a skeletal muscle cell. And there are hundreds, thousands of examples like that for all kinds of diseases. So this, we think, will be a really valuable resource. And CZI is not building this. We are supporting the development of this human cell atlas. It's led by Aviv Regev and Sarah Teichmann, two incredible computational biologists and biologists at the Broad Institute, and EBI Sanger. So they, and now hundreds of labs that have sort of gotten together in a grassroots effort to build this resource, are all trying to figure out how do we, as sort of scalable as possible, high throughput as possible, characterize all the cells in the human body, and then organize that data set and make it widely available to researchers. So at CZI, we're involved in this by funding groups, experimental labs that are developing new technologies and new ways of making these kinds of measurements. We're involved in infrastructure, building data infrastructure to host all of these data. If you're going to do machine learning and computation, you have to have the data be openly available for people to do that computational work. So we're working on that. And then we're also involved in learning and better understanding what are the computational challenges and different kinds of new ways of using different machine learning techniques to better understand all of the data that we get from these measurements. So I just wanted to show one example. One thing to say about this community is it's incredibly open and collaborative. And sort of in that spirit, I'm going to highlight work of another group. This is not our own work, but this is work from Sten Lenarson's lab. How does this work? Oh, great. This is work from Simone Codalupe and colleagues in Sten Lenarson's lab. And it's using a method called image-based transcriptomics that I think is incredibly cool. And again, I really want to highlight the work of others, but this is a group we're collaborating with Sten's lab, as well as a handful of others that are working on this method. So if you think about, you want to build an atlas, you need to know the things about all the different cells, and you also need to know how they're organized in space. Because a lot of methods, for example, single-cell RNA sequencing, this is where you sequence patterns of gene expression in individual cells. The way you do that is you take all the individual cells out of, say, a piece of tissue, and then you basically grind them up. And that's great. You can then count all the different, the expression of all the genes, but you lose track of where the cells came from. So it's sort of like a census, whereas if you want an atlas, you need to know where the cells were. So there's been, over the last 20 years or so, and especially over the last couple years, a whole bunch of new technologies that are called image-based transcriptomics. And the idea is to measure the same thing, measure patterns of gene expression, but actually do it in the cells, in the tissue, in the body where they were. So obviously not in a live body, but if you in this case, we're looking at a mouse brain. So the idea is to take tissue, and then you build these fluorescent probes. So they're basically little markers that go in and bind to a particular gene that you're targeting, and ideally many genes at once, and that's some of the cool tricks that are being used. And then you have a little fluorescent probe, so that when you would take a picture, you see all these little spots light up, and those spots correspond to individual RNA molecules where that gene is expressed. So this has been done, it's called FISH, fluorescence in situ hybridization. It's been done for 10, 20 years. What's just happened in the last couple years is the ability to capture these images and look at hundreds of genes simultaneously by multiplexing. And this has totally blown up the ability to do this in a really exciting way. So here's an example. I love this figure. First of all, this is super recent. This came out like 2 weeks ago. So it's really exciting. It's also probably, in terms of like information density, one of the most information dense figures I have ever seen. So we're looking at a really large fraction of the mouse brain right now. And you see how we've zoomed in on these tiny little squares. Every single one of these spots and every single one of these pictures is an individual RNA molecule, and the color is based on a particular gene that was found to be expressed at that location. So in one image, we have an incredible amount of information about both the location, the sort of subcellular organization of gene expression, and then also the patterns of gene expression, how they vary over, in this case, an incredibly rich and dense tissue, the brain. So the one thing to highlight here is there are really multiple ways in which machine learning and computation is critical for understanding these data. The first thing to say is that the images I get out when I do these experiments, they don't look like this. They're not pretty with nicely colored spots. They're just raw images, a little bit like what Jeremy was showing on the left of his figure. So we need to do analysis and computation to figure out what was the gene that was expressed at each of these locations. That involves doing a lot of computation. It's really important to get that right. After you've done that, you now want to know where are the cells. So we don't want Nick having to draw a circle around every single cell, which he would also have to do here. So there are also algorithms, and in fact, people are using new machine learning methods for image segmentation to go in and be able to automatically define the boundaries of individual cells, like shown in the lower right, where there's a little white circle around each of those cells. So in fact, there's a lot of overlap with some of the methods Jeremy described, and this is a really exciting area of machine learning that hopefully will make it so Nick and people like Nick will never have to draw circles around cells again. Then once you've done that, that's sort of basic data processing. That just tells you where is every gene and where are the cells and which genes are expressed in which cells. After that, there's all kinds of analyses, both unsupervised and supervised, that you want to do on top of these data to really understand what are the patterns in the data, what are the spatial patterns of gene expression. Lots of organs have patterns of gene expression that vary in really rich and interesting ways. You can imagine this within normal healthy, or this happens within normal healthy functioning tissue. Also, if you're looking at, say, a tumor and you want to understand how does patterns of gene expression change within the tumor, what are the differences between tumor cells and non-tumor cells? This is exactly the kind of technique we're going to need to be able to do that kind of high throughput and spatial characterization. Final thing that again, back to my neuroscience background, I think is super cool about these measurements is that they could potentially be combined in completely different experimental assays. So if we go back to thinking about the brain and the microscope, you could, not literally in this same mouse, obviously, but in a different mouse, you could, in the same organism, same animal, measure the pattern of activity of all of these neurons. So we could look at which of these neurons were lighting up or not while the animal performed some behavior. And then in the same mouse, we could go in and characterize patterns of gene expression and ask questions like, when the mouse learned to do a particular behavior, is that related to changes in the expression of certain genes? And what does that tell us about the basic mechanisms of memory in a functioning sort of normal organism, but then also what might happen when they go awry? So these measurements, the ability to do them, the machine learning we need to analyze them, and then the ability to combine them with other kinds of measurements, I think will be truly transformative for both basic health biology as well as understanding of disease. And it's been amazing to be collaborating with folks like Sten and working on these kinds of projects. So really looking forward to hanging out tonight, and thank you all.
All right, so a lot of excitement, maybe even a lot of hype here tonight in the room, and I want to kind of bring us down to earth a little bit, and then we can go and have a drink and drown our sorrows from what I'm about to tell you. So there's a lot of data with a lot of information in it, like you just saw, and we absolutely need computers to analyze it. Because they do better than those student interns, because there's no way a person could go through all this data and find the patterns. And we're excited because there are some really promising new methods available for doing this. But I think we need to slow down a little bit and ask, are we using the right methods for these problems? Where are the limits in what they can and can't do? And so I just want to share a couple thoughts on that. One is related to interpretability. So as I mentioned, what I'm most excited about with some of these models is not just that they can predict what type of cell it is or that it's dying, but why. What was that cell doing? How did it look in the microscope? What genes was it turning on and off that tell us that it's dying? Because that is going to teach us about cell death. That's what we care about here at Gladstone, the basic mechanisms behind diseases so we can understand them and try to fix them. And unfortunately, some of the AI methods that perform best right now, that make the best predictions, that beat the chess players and outperform these student interns, while I think are much more interpretable than early iterations of those methods, I think we still need to work on this question of interpretability. It's not enough just to have a good prediction. We at Gladstone want to know why. We can't have a black box. So that's number one. Number two is reproducibility, and that's related to the fact that you could do really well with a certain approach to analyzing this kind of data, and then someone else tries it on another data set and it bombs. And that is happening in this field of applying AI to biomedicine, and it's happening in other applications of AI, and there's a couple of reasons. One is that you might have over-taught the computer how to do a particular task or how to really find the patterns in your data set. And in that overfitting, you've created a situation where your algorithm now doesn't generalize well to other situations. This is not specific to AI. This is a fundamental problem in statistics. You want to fit the data, but if you really want to generalize outside of your data set, which is our goal here, to learn something bigger than our own data set that can have a big impact, you don't want to overfit. And so I think we need to think about, would these algorithms have a tendency to be very good at finding patterns? Are we being too good at that? The last thing is that these algorithms are super complicated. So these guys were very humble in presenting their work, but in getting those images segmented, drawing those circles around them, or in predicting if those neurons were dying or not, they had to tune a bunch of different things. It wasn't just like run the code and out comes the answer. They had to make a lot of choices in that code. And there are so many choices in these algorithms that we make that it's almost impossible to turn each knob one at a time to try all the combinations. So what's happening right now has been described by some in the field as alchemy. Someone gets it to work really well with a certain set of choices, and then they go, oh, this is the secret sauce. You guys should try this too. And people are copying that. And sometimes it works and sometimes it doesn't. And there isn't this sort of, we forget to be scientists and do all the controls and check all, and be rigorous and check that do we really know why it's working or why it's not working. And so this hype and hearsay is a problem. And so what I recommend, and these three things are not specific to biomedicine. They're general problems with AI. And what I always recommend when people come and go, I've heard this AI stuff is really cool, how do I do it? I say, have you just calculated a correlation between this thing and that thing? Did you see, were there any patterns in your data to begin with? Before we start doing this very complicated thing on the computer, where literally the computer's doing stuff that people don't understand. So I'm sort of trying to take back the reins from the computer a little bit and use our own brains. To conclude, I want to say a couple things about the limits of AI that are specific to biomedicine. One of them is that the stakes are really high. Deepak set this up at the beginning, talking about his family members that aren't well. I'm not well. I've been in the ER in the past week for my medical conditions. If we're going to use these methods, we want them to work really well. And this is really evident when you think about letting a computer make a clinical decision, like whether you should give a drug or not, or whether you should make a choice in the operating room or not. And there's arguments for doing that because, as you just heard, the computer can do better than even a well-trained expert a lot of the time. But do we really trust them? You know, and it's, I think, the same even in science. The decision isn't as imminent as like, should I cut this person here right now or not? But even in our research, the stakes are very high, as Deepak described, and so we want to do the right thing. We don't want to be carried away by hype. Another reason that that's specific to biomedicine that I want to suggest that we think a little more carefully about AI and how we're using it is that AI does really well when you have a lot of data. And you just heard from these guys, I had so much data, I didn't know what to do. But let me remind you about the stats lesson at the beginning. 2 objects, picture 1 and picture 2. That's 2 pictures. And I said you're going to measure a lot of stuff. There's a ton of pixels. But what you need actually for these algorithms to do well is lots of pictures, lots of examples of your sister and lots of examples of your son. It's not so important that how many pixels are in each picture. And just to take that analogy then into biology, unfortunately, what we have, and we kind of forget because the data set is so big and we're really excited, we actually have the equivalent of so many pixels, but we actually don't have that many pictures to look at. So we are measuring everything you can possibly imagine on these cells, but it's just one slice of a mouse's brain from one mouse. What you really want is a lot of mice and a lot of slices and to really learn those patterns. And that's possible theoretically, but from a practical point of view, I've seen very few biological data sets that have lots of examples. They usually have a few examples and a lot of stuff measured. And that is not the ideal situation to be applying AI. And it amplifies some of these problems of overfitting and generalizability. And then just getting a little more subtle here, the algorithms really expect that the faces are independent pictures. But in biology, often they're not. It's actually the same cells just in a second round of the experiment. And they're not really independent from each other. And that can also contribute to the overfitting. And we've had that problem, and we're in fact in an argument in the literature right now with a colleague about whether when we were predicting if these disease mutations affected particular genes, were we really, and we said we could do it really well, did we actually evaluate that with independent data? Well, ideally you would, but there's only one human genome. There is no independent data for us to go out and invalidate that on. You can't take another picture. And so, some of the things that one would ideally do when applying these methods are not possible, or at least not easy to do in biomedicine. There's also some just implementation challenges, like you need to make your biological data set equivalent to pixels. And sometimes that's easy to figure out how to do that, and sometimes it's not. And also there's massive computer power needed to do what you just heard about today. And that's expensive, it's hard to do, and it's something that I'm spending so much time trying to figure out just how to actually make this stuff work. So should we even be using AI here at Gladstone or in biomedical research? Of course we should. You heard a lot of promise, you heard a lot of success stories today. Why were those 6 stories exciting? Because they told you that you can do something by fitting a model that you can't do just by looking through the data, leafing through it. So what do models do? Even if the models are bad or they're not perfect, models allow us to predict things, and they allow us to understand mechanisms. And so I still think we should be fitting models to biological data. I just think we need to think very carefully about what models we're using and what their strengths and weaknesses are, and not get carried away by too much hype. So that's my personal take. I'm very excited despite all those caveats about these approaches. And I think the solution to the fact that this is hard is not to throw up our hands and give up, but to really bring together the teams that can think deeply about how to do this right. So that's what we're trying to do with this new initiative in data science here at Gladstone. Get these interdisciplinary teams that have the real expertise in computer science and in statistics, and have them sitting together in the same building, in teams, analyzing this data with the people who are generating it and who are thinking about the biology. And I think that by bringing those together, we can avoid some of these pitfalls and really make a difference. So I want to ask the 2 Jeremys now to join me on stage, and we're going to do a little Q&A. We'd love to hear your thoughts.
I'd like to thank all the speakers for really insightful comments. And we have time now for some questions from the audience. Yes.
Yeah, hi, so I'm Phil Nelson. I'm one of the Google people on the other side of this. I just want to make 3 points. One is your talk was... Oh, hi. So I'm Phil Nelson. I'm from the Google team that's been working closely with Gladstone. I just want to make 3 quick points. One is thank you very much for that talk. A lot of the challenges you're talking about, we have AI researchers hard at work trying to fix. Secondly, doing things like transfer learning from consumer data to see if we can apply it. But the bigger point I want to make is I want to just thank Gladstone. Like when Mike and I started this team about 5 years ago, and Gladstone was our first partner, and it's been just an incredible relationship. You guys do amazing work, great controls, such great scientists. So it's been a fantastic collaboration, and I'm really excited about the projects we have going forward too. The in silico labeling was great, but there's a queue of work ahead of us that we're super, super excited about. So, you know, to all the people here for the President Council, Gladstone's an amazing organization, so thank you.
Well, thank you for that. It is true that, you know, we do deeply believe that the way that we're going to make the most disruptive insights and creative insights is through creative collaborations. And, you know, this example with Google is one of those. And in fact, the director of the National Institutes of Health, just a few weeks ago after this story was published with this Google collaboration, highlighted it in a weekly blog indicating holding this up as the prime example of that sort of interaction and using AI in medicine. Yeah. Just the beginning. That's right, it's just the beginning. Yes?
Assuming there's other people who are as smart as you are and with your resources, but unbound by ethical concerns, is there a risk that more problems could be created than solved by this new era?
I think that's Jeremy.
It's a Jeremy question, but which Jeremy? I think that's a super important question, and one that definitely affects me quite personally. I think it's a big concern. I think there are, as I'm speaking for myself now, definitely not CZI. There are concerns at many dimensions and potentially many timescales. Sometimes in the news you see things about, I don't know, sort of crazy ideas like robots taking over the world. I think AI is getting better, but we're still really far from that. In some ways, the things that I feel most concerned about in the short term are around things like bias. One thing related to this and the issues that Katie brought up, these methods they reflect the data that you put in. And especially for when it comes to AI algorithms that are making important decisions, whether it's affecting your health or making recommendations to you that are going to affect your personal life, when those recommendations are biased because the data that went into those algorithms are biased, they only included examples of certain kinds of people or certain kinds of disease, and then are making biased recommendations as a result, that's a real challenge and I think a real concern. So I am very much in favor. I think there's a handful of groups out there that are thinking very carefully about how to have sort of more ethical considerations about the use of AI from the start. I think those are great initiatives, and there should be more of that, and it's really important. I think that stuff is more short-term, a big concern. The sort of robots taking over the world, I think we're definitely pretty far away from that.
Here and then Clark. Yeah, go ahead.
Hi, I have a specific question about a certain type of application of AI, and especially when it comes to neurodegenerative diseases. So maybe this is a good question for Jeremy number one. So the question would be, has there been interest or headway in using brain organoids to do drug discovery? Because using organoids, which seems to be a much better simulation of the complexity of the human brain versus obviously disassociated cell cultures, plus machine learning to do drug discovery, is this something that you guys are looking into or have interest in? If not, why not?
Yeah, absolutely. We are definitely interested in using organoids, and we have several projects that we're currently working on where we're trying to develop organoids.
Can I just interrupt for a moment and maybe explain to the rest of the crowd what an organoid is?
Please, yeah.
So this is one of the real wonderful breakthroughs also in the last few years. Many of you know that here at Gladstone we developed the technology to make stem cells from any one of your blood or skin cells, make them truly like embryonic stem cells. And we could grow initially those in a monolayer in a dish and study them. But now for a variety of different organs in the body, we can grow them in a three-dimensional shape that mimics some of the 3D features of the actual organ. And so different cell types will come together within an organ and mimic that. And that allows a better model which to study than a monolayer of one cell type. Here you may have 3 or 4 different cell types that make up an organ.
So yeah, that's a great introduction. we've actually developed a robotic microscope that can image not most of the images, or the image that I showed you, but most images we take in the lab are two-dimensional images. Essentially, we're looking at one plane of a monolayer of cells. We've recently developed a microscope that can image in 3 dimensions, and actually, since we do it over time, we call it a 4-dimensional microscope. We're just submitting a paper on that right now. So we've used that with we've used it with slices of brains from mice. We've used it with slices, human primary brain samples as well. And we're working with collaborators around UCSF to use organoids with human tissue and the mix of the human tissue. But I do want to say one disclaimer on this. Although they are better at replicating certain features of neurodegenerative disease, they're more difficult to analyze. And so we run into this big problem. I mean, we're able to generate large data sets of 2-dimensional images, but every single sample, if you have to take 100 images to get the 3 dimensions, it makes your problem 100 times harder. So generating large data sets of 3-dimensional images is actually currently impractical. And I actually do a lot of 3-dimensional imaging now. I have a bunch of other projects where I image zebrafish, live zebrafish as well. And I can't imagine at this point generating large enough data sets like Katie was talking about, where you can label them sufficiently that you can use artificial intelligence with a level where you feel comfortable with it. So it would take a substantial amount of effort and resources to do that. At the same time, the way I like to think about it is you do the imaging on your 2D culture, and you use artificial intelligence to inform new hypotheses on what you think is going on, and then you test it on the organoids, and then you test it on more complex things, and then you don't mind drawing circles around the neurons to measure it.
Can I add one thing?
Yeah.
Yeah, just a quick addition to that. I'm also using organoids in work where we're trying to understand psychiatric diseases and specifically to understand the parts of those diseases that start during development of an embryo. I think for that, the organoid is very good because the cells in them are really more like an embryo's cells. And for studying a disease that comes on at the end of life, you guys, I know, have the challenge, and this is true for studying heart disease as well. If you want to study how the heart fails, you need to make an organoid that looks like an aged heart. And Deepak's done some amazing work trying to make cells age more and to make the organoids more realistic. But I think this is one of our main challenges going forward.
Yeah, but your question is a good one, and that is the future, and it's a lot more data, as you heard. But we're here to imagine what we can't imagine today and see. Clark?
So in working with artificial intelligence, what are the most sort of unexpected, surprising things that you've come across?
I can start. Maybe we can each give a short story. What has surprised me is that the problems I thought were hard, because they're hard for a person, are sometimes easier for a computer. And the ones that we think are going to be easier, the computer actually has trouble doing. It's very interesting. So even though these algorithms are mimicking, we call them neural networks because we wrote the code so that the way it communicates is like the way we think the brain works, there's something fundamentally different about the code from how our brain actually works, pointing out that we don't actually understand how our brain works. So for me, the biggest surprise has been that literally in my lab has had 2 of these problems about these mutations and which gene do they mutate. There was one that I thought was going to be easy that we've done only so-so on. And then when I told the postdoc, stop working, don't do this, it's going to be a total failure, that one worked great. So.
As I told you, I'm a biologist, and kind of the eureka moment I had with artificial intelligence was when we trained some artificial intelligence to tell whether the cells were alive or dead. And then, as Katie mentioned, a lot of times when you do this happens actually a lot. People will do this, and then they'll try it on a new data set, and it won't seem to translate. But what really happened for me was we trained a model, and then we tried new data sets, that the artificial intelligence had never seen before. And it was still able to predict or say whether a neuron was dead or alive. And that's when I knew that this had arrived. It works. And it doesn't work equally for every data set you use, and there's definitely going to be differences. But to see that it translates and that it's seeing what a dead cell is, and that's when I was like, I'm in. This is great.
Maybe continuing the theme of sort of what's the difference between neural networks and the real brain, I think something I'm continually surprised by and super curious about is that there are actually tons of things about the real brain that are really interesting and unique and not well understood that we have never tried to incorporate into the models that we use when working with neural networks. Just to take one example, neural networks, the brain has a lot of different types of cells. We don't even really know how many there are, but it's definitely in the thousands. So in your brain, there are thousands of different kinds of individual nerve cells that have totally different functions and different properties and different biophysical properties, different morphologies. They look different. In a neural network, if we're being generous, there's like 3 or 4 different kinds of units or neurons in an artificial neural network. That's really interesting because if we used thousands of different kinds instead of 3, they might do a lot more interesting things. It's a totally open and not well understood question. So that's one possibility. The other possibility is that the thousands of cell types that we have, it's an artifact and they don't actually do anything that interesting. I don't think that's true because I think there are a lot of things still that that human brains, even brains of simpler animals do, that are truly stunning and are things that our current artificial approaches are not capable of. Whether we could learn from aspects that make sort of actual biological systems unique and then translate that knowledge back to the design of artificial systems, that's something I'm very interested in, and I'm continually surprised by the possibility.
Great.
Then they might really take over the world, yeah. Well.
Sounds like we're safe for a little while. Clara? Maybe for everybody else.
Sorry. I'd like to hear about more successful applications, either at Gladstone or other institutions where, it's really been successful, leading to some great discovery that, that other scientists have been able to build on, just so that, we can all understand more broadly, how this can really change the world.
You said reproducible and interpretable?
Well, just in ways that really get over some of the limitations.
So I mentioned disease non-coding mutations. They're outside of genes and figuring out whether they're functional or not. I think we're making good progress there. Not just my lab, but lots of people are working on that problem. Machine learning's been very useful there, and the reason is that there isn't a simple signature, and you need a computer that can find that kind of complicated pattern. Another area of biology that you heard about today was in analyzing images. We still have a long way to go, but I think image analysis is a huge opportunity for AI, and one where we really need it. Like, there's just no way, again, to do that without a computer. Because you need something high throughput and you need something that can detect on subtle patterns that maybe once off the human eye can detect, but to integrate all that information, the computer is very good at that. In clinical decision making and predicting outcomes, for a cancer patient comes in and you sequence their genome and you keep track of their diet and what medications they've been on, predicting whether a treatment will work or not, whether it'll have side effects or not. Way before we called it AI, 20 years ago, statistical prediction methods were working pretty well there, and they're doing much better now. That's one of the ones where the stakes are really high, as I mentioned, and so I don't know if they're good enough yet. I'd like to see them do better, but they're doing much better than they were. So those are a couple that come to mind. Another one my lab is working on is not about the human cells in our body. Deepak mentioned I work on the human microbiome, so all the bacterial cells in our body and other microbes. And we want to try to associate them and their genetics with diseases. And the problem there is a little bit like Jeremy described, that there's so many cells in the brain and we don't even know what kinds they are, but it's like a trillion times worse because most of these bacteria no one's ever studied before. And so we can't use a method that tries to understand the microbiome by referencing back to our knowledge base. We need to just learn the patterns from the data It's let the data talk to us and tell us what the patterns are, because we don't know enough to even know what patterns we're looking for. And machine learning's been incredibly useful there. So that's a couple from me, and I don't know if anybody wants to add any.
Go ahead. I have one,
Yeah.
but you can go ahead. I would just add in I've seen there's been a number of reports lately where we can take skin biopsies and train using pathologists to label pathology slides, feeding that into artificial intelligence to tell whether a specific there's a great paper on melanoma to see whether a melanoma is malignant or and I think that's amazing, and that's actually similar. We're trying to apply similar stuff to our neurons in culture. Yeah,
That's a great one. I was just going to mention behavior. So really broadly construed, whether you're talking about a worm or a fly or a mouse or a human, being able to statistically, quantitatively describe behavior is really important for study of basic function as well as for disease. And I think there's been some tremendous work in machine learning to make that process more automated. The last thing you want to do, again, is sort of take a video of a behaving mouse and then go in by hand and literally, every frame of the video say, now the mouse is walking, and now the mouse is jumping. Do mice jump? Now the mouse is turning, or now the mouse is talking. Instead, you want to somehow extract that from the data and have it be as rich as possible. And some really cool methods have been developed to do that. One recently I know about this guy, Alex Wichko, who was at Harvard and is now actually working with Google, maybe knows these guys, was basically developed a method. He used something called the Kinect, which was from a Microsoft video game system, to capture 3D views of mice running around, just free behavior. And then he used some really cool machine learning methods to not try to label every frame as being one of 5 different behaviors, but just let the model sort of learn the structure of the data and just describe all the different kinds of behaviors that mice tend to be engaged in. And then he was able to take that method and now treat the mouse with a bunch of different drugs and other sort of potentially behaviorally affecting chemicals, and was able to immediately see differences between different drug treatments that had never been seen before, because all people had been able to do was ask, does it make the mouse run more? Well, no, but it does change the behavior, and he was able to see that by using this new method of uncovering structure in behavior. And that's good. I think you can imagine doing the same things for humans, and that's super interesting.
Yeah. So it's early days, but clearly there are some examples, but you can imagine so many more different things. Yes?
So recognizing both the potential of this technology and its pitfalls, and I think they both exist, 10 years from now, looking back, putting research aside, where do you see the biggest potential for breakthrough therapies or medicine, where the medicine is changed by the application of these tools?
By artificial intelligence.
Yeah.
So I mentioned this a little bit previously. So I think of artificial intelligence as informing new hypotheses for how we should take our science. So I feel like science kind of gets into these little bubbles of what we think science is going on in science, and it's always wrong. We know that there's parts of truth, but we know that we're just looking in a bubble, and then eventually that bubble gets broken open, and then we see that, oh, that was silly. The Earth is round, and we revolve around the sun, right? So what artificial intelligence, I think, can do is give us hints that we're not being able to see with our eyes. Our eyes are imperfect, and if we can find an interesting result with artificial intelligence that we wouldn't have otherwise seen, then we can use our conventional methods to figure out what's going on there. So I think we're going to learn a lot about biology. There's a lot about biology that we don't understand, and we don't even know where to start. So it will inform new hypotheses and break open new avenues of research that I can't think of right now.
One I'll highlight, just been on my mind recently, because there's been some really cool recent work on it that I think is a space kind of ready to blow up, is the use of machine learning in structural biology. So at the end of the day, both a lot of the things that we think about in terms of characterizing how biological systems work, and also the way we think about intervening with them involve proteins and other kinds of small molecules that have complex structures and interact with each other. And if we want to design drugs, if we want to understand how to make drugs do certain things, we need to understand those structures and why they behave the way they do. This is something that is incredibly hard to get one's head around. Structural biology is incredibly complex, and whether it's about predicting the structure of proteins from sequences, which is a sort of famous problem in structural biology, or designing novel proteins, designing proteins that have functions that have not been discovered by biology, but could be incredibly important for human health. Just in the last few years, there's been some really interesting examples of people using machine learning to kind of really reframe this problem and take a very new approach to some very old problems. And I think it's just very early days of this. It's just starting. But from some of the things I've been seeing, it's a very interesting opportunity. And I think in a world in 10 years where a large fraction of the molecules we interact with are designed, I suspect that will be driven by new advances in machine learning that let us do that in a more systematic and automated and data-driven way.
So maybe just to clarify, structural biology may not be obvious to everybody, but the proteins in your cell are the ones that do all the work. And so the proteins have a shape. And what we're now beginning to finally understand is that a certain shape of a protein, and that's its structure, is what's responsible for its action. If you change the structure of the protein, then it doesn't work right. And a lot of drugs that we all use for a variety of diseases are going in and changing actually the structure of that protein, and that's how they work. We never really understood that before, but now we're able to, with X-ray crystallography and other types of electron microscopic approaches, finally understand the shape of our proteins that are doing all the work. And it's really biology is becoming more physics by virtue of that. So.
Yeah, from the perspective of therapies, I think one of the biggest opportunities is actually in understanding chemicals better also. So you can, in the same way that I described taking a protein profile or gene expression profile and thinking, can I make this look something like pixels in an image? You can describe chemicals and their structures and how they interact with proteins by encoding a bunch of bits on the computer. And then I think we could really understand what kinds of chemicals there are and how they relate to each other in a completely different way than we do right now. And I think that will have huge potential for repurposing drugs, for predicting which drugs are going to work. Right now, the way that drug development mostly works is by brute force of trial and error, and some good medicinal chemists who have some intuition about this. But I think the computer could be incredibly useful there.
I know we have some wine waiting for us. Let's do one more question, and then we'll...
What you're doing sounds very exciting and very far away. What you're doing sounds very exciting and very far away in terms of coming up with a therapy for ALS or Alzheimer's. Can this, and is this being used to do something that sounds like much more simple? Like, why am I having a radiologist to read my X-ray for my broken bone or my mammogram or my echocardiogram when it sounds like the computer might be way better than at it?
Yeah, awesome point. I couldn't agree more. I think, I don't know exactly why that's not happening, but I think one of the reasons is about data access. So to develop the algorithms that could do better than the radiologist, we would all need to be able to see those images. And this is just something I care about, and I know Jeremy Freeman does too, is a lot of the data in science and in biomedicine is very siloed and doesn't allow the people who could develop that algorithm to work with the data. So it's totally possible, and it's amazing that it's not happening right now. And maybe this is just my own personal take, but I think part of it's like some siloing or some territoriality.
But it is starting to happen, and people are testing that. So, for example, like you said, radiology is a great example. Already, radiologists can be anywhere in the world reading your X-ray that you get when you go to the ER. They could be anywhere, and radiologists who with experience has looked at 1,000 X-rays of somebody with a certain thing or a CT scan, and based on that experience, they can see a pattern. But what you're saying is that instead of 1,000, somebody could have experience, a computer could have experience with 1,000,000. And that is getting tested right now, and that will be the future, and it's not that far away. For echoes, you also mentioned for heart disease, and particularly I'm a pediatric cardiologist, so children are born with malformed hearts that have distinct patterns. And sometimes, particularly by fetal ultrasound, if a mother gets a fetal ultrasound outside of San Francisco or Boston or Houston, then they've got a good, 80% of the time that pediatric cardiologist will make the right diagnosis. Anywhere else outside in a smaller town, it's only about 50%. So 50% of the time, they'll miss a congenital heart defect in a kid. And so we're actually, one of the investigators here is trying to use machine learning tools to take those images, feed them into a computer so that anybody anywhere, democratize that knowledge and that experience so anybody anywhere can do that. So you're absolutely right. That's really coming soon on. And then what's more immediate even for human disease and discovery, the work that was recently published from Steve Finkbeiner's lab with Google folks was related to Huntington's disease. And so seeing patterns in human neurons that we couldn't otherwise see, and then now it's ready for drug discovery to correct those patterns. So that's happening now. So I would imagine very soon we'll be seeing drugs that are ready to move forward into trials that were discovered based on their ability to correct some pattern that we never would have seen had it not been for those approaches. So, well, I want to thank our panel for wonderful talks. I hope that was informative and enlightening, and you got a sense of what lies on the horizon. I do want to, before we break and go out for some wine and a reception, there are a number of Gladstone faculty here in the room. If you guys wouldn't mind standing up real quick so that people know to find you. If you have additional scientific questions, they will be a number here left earlier. But feel free to flag them down. And as you drink your wine. Well, I want to leave you with one thought, and that is that as you've heard today, we have great challenges in front of us, but we have great opportunity, and there's never been a greater need to go along with that, the opportunity and the challenges. So I've never been more excited than I am right now about our ability to finally understand human disease in a way that is going to allow us to create new therapies for, as I mentioned earlier, these intractable diseases. So I appreciate all of you guys coming here tonight, learning more about Gladstone, supporting Gladstone, and I think together as partners, we will tackle this. So thank you very much.